Hello, and welcome to this workflow demo on high-throughput sequencing for complex disease research. My name is Cora Vacher. I'm Associate Director for Human Genetic in the market segment team at Oxford Nanopore Technology. I'm delighted to be joined by three amazing speakers who will share more information about their sequencing workflow. You heard the plenary from Kathy and Natalie this morning. And after, we'll have time for a quick Q&A session. So this is an on-site only event. We will be taking questions from the floor, but we will not be taking questions from the event app. I will introduce the speaker in a few moments, but first, I wanted to present a brief overview of the real-life example that Oxford Nanopore is doing for complex disease and the solution we have for large cohort. So understanding the genetic architecture of complex disease has always been a numbers game. But now, with Oxford Nanopore, it's really possible to get much richer insight on large group of participants. For example, long-read sequencing typically identify twice as many germline structural variants that you would get with short read. And obviously, you also get, with Oxford Nanopore, the methylation information simultaneously from your genomic information. So it's not surprising that there's now a number of large projects that are using our solution to really understand their population. One of the first one I wanted to present is PRECISE. So it's a project, an initiative that we have in Singapore. The team there is sequencing 10,000 genome from the representative from the multi-ethnic Singaporean population. They're also really improving the pangenome by doing 100 telomere to telomere sample. Our speaker will present the data and the workflow from the 22,000 sample from the NIHR bioresource, so I'm not going to go in detail over that. Last year, we announced that we're doing 50,000 sample with the UK Biobank. It will make it the biggest single cohort that has, at the same time, structural variant and methylation information. And the last plenary before the break that you've heard by Kimberly Billingsley really showed that the team at the NIH Center for Alzheimer and Related Dementia is doing with about 4,000 brain sample and brain tissue. So the sample in large cohort and biobank can be really varied. I mean, if you think about human sample, most of you will think blood, saliva, buccal swabs. As you saw with Kimberly's presentation, we also have people doing cells and tissue at scale. And we're seeing a growing number of people using cfDNA to try and identify markers. And so our P24 and the A-series tower are really much the ideal production tool for generating a lot of genome per year. And by a lot, I mean we can generate about 2,500 genome at 30x per year. That's about twice as much as you would get with a Revio, which does, I think, about 1,300 samples per year. And it's also way more flexible than, for example, what you do on NovaSeq, because you can do one flow cell at a time. Our Epitome desktop application offers a very comprehensive analysis of the human genome in a single workflow, really simplifying the investigation of human genomic variation, including SNVs, SVs, methylation, as well as copy number variation and short tandem repeat. Epitome provides secondary analysis, including annotation file and report for all of the mentioned variants, as well as SNV phasing block. The visualization of the map read and the different track are available either on Epitome or you can open them on your favorite genome browser. So in summary, we have resources to really help support the production scale sequencing, including really robust output and streamlined protocol. We have dedicated support team, including field application scientists on-site and online to support your project 24-hour, and customer and technical service to help you. Now, investing in training might be the best way to start. And we offer online nanopore learning course, but we're also on the community, but we also do on-site training. So now I'm delighted to introduce our three wonderful speakers, who are Alimat Oyaboye, Matilda Tierney, and Cathy Stirrups, whose fantastic plenary presentation you heard earlier. So I'm not going to do them in the order. I'm going to do them in the order I've got them there. So as a brief reminder, Cassie leads the NIHR Bioresource sample team. She also oversees the feasibility and provision of sample to research group and manages the bespoke project, such as the whole genome sequencing or RNA sequencing to assess the translation of this technique into health system. Alimat is a research lab technician at the NIHR Bioresource in the UK. In her role, she contributes to the rare disease RNA phenotyping project, which aims to recruit about 1,000 patients, as well as the long-read project, focusing on the sequencing of those 22,000 sample patients that we're presenting this morning using Oxford Nanopore sequencing technology. And Tilda is a development scientist based at King's College London, where she supports the NIHR 22,000 sample project, completing a high-throughput whole genome sequencing workflow using Oxford Nanopore sequencing platform. Welcome on stage, ladies. Okay.
Hi, everyone. So I'm going to be talking you through how we took our project from the pilot phase to sequencing 384 samples per week. And we have a disclaimer that all ONT products are research use only. And yeah, so we took our project from the pilot phase to sequencing 384 samples per week across 2 labs and 8 proms. We started by defining our project goals, and that involved setting realistic expectations early on and balancing our desired N50 with our yield to get the 30x genome coverage that we're looking for. Next, we looked at our samples, and DNA quality drives everything. And this had us looking at our source materials and evaluating the extraction method. And sorry, I can't see the, thank you. It also had us tracking sample degradation, and we also made sure that we carried out thorough QC on our samples, as well as developing or using strategies to improve our fragment length and fragment distribution. Next came optimization, and this had us looking at fragmentation strategy, exploring DNA cleanups, and we also tested the manual versus automated workflow, and we also brought in bioinformatic feedback early on in the project, which allowed us to balance our wet lab output with computational and And next, we went to the implementation phase, and this had us deciding how many rounds of implementation we're looking to carry out, how many technical replicates to include in this round, and also we had different lab techs doing these implementation phases to introduce buyers and also to account for buyers during sequencing. We also looked at the PromethION capacity and also wash and reload strategy to further stretch our resources without compromising on data quality. Next, we looked at our QC, and early on in the project, we introduced LIMS, and we made sure that we had quality checkpoints from DNA extraction to sequencing, and also we considered the operational sustainability of our projects, and this had us considering the time and manpower, the lab space, and the workflow, and also we made sure to use every fail run as a feedback loop to help us prevent repeat issues, and this allowed us to scale our projects and also to have confidence in our data.
Thank you.
So we thought it would be useful to kind of take you through the day-to-day effort that goes into completing high-throughput whole genome sequencing using Oxford Nanopore sequencing technology platforms. So at KCL, the aim in the production phase of this project is to complete 288 samples per week. 144 of these are blood samples, so they're slightly higher quality, and only require one wash and reload, and the remaining 144 are saliva samples, which require 2 wash and reloads. So kind of the routine lab tasks I've categorized into normalization and shearing, library preparation, sequencing, and other. And on the slide at the minute, you can see kind of the distribution of these different task categories throughout a standard week at KCL. So KCL is one of the sites where we're doing this sequencing, Cambridge being the other one. And I think it's good to highlight here the kind of more administrative tasks that are involved with the lab work. So these are tasks that are kind of completed around the routine lab work, kind of as and when, and involve kind of sample management. So that's kind of receiving samples, organizing the samples and picking the samples, reagent and flow cell management, data entry into our internal LIMS systems, troubleshooting any unexpected experimental outcomes, and also liaising with our collaborators at Cambridge and with O&T about the progress of the project and kind of any upcoming changes. So to complete all of this work, we employ the use at King's of 5 research assistants, 1 Hamilton pipetting robot, which we use for normalization, 1 fast prep, this mechanically shears our DNA, 1 dream prep, which is a TCAN robot that completes kind of automated ONT library preparation, and finally 6 Prometheon 48 machines. I think it's important, again, to note here that only 24 of the 48 positions are only ever utilized at any one time in each prom.
Okay, thank you. Hello again, everyone, and I'll just take you through a few of the things you might not consider when you're thinking about setting up a really large-scale project. So the first one's to do with the infrastructure in the lab. This has been a bit of a steep learning curve for us because we've had to think about everything from making sure we've got enough power supply, enough sockets and circuits to power all these high-throughput machines and DAUs and monitors and things. The other big thing was temperature. These Prometheans kick out a lot of heat. So as soon as you have more than 2 of them around, things really start to hot up. So you need an aircon that can cope and an airflow around your lab. Otherwise, it can get easily up 35, 40 degrees, and then the lab techs start complaining, let alone the sequencing. The other thing, these projects, they make a lot of data, so you need to be able to have plans for keeping all that data, as well as the actual analysis, the downstream analysis. And as Tilda mentioned, there's quite a lot of other lab equipment going on just to do the library prep that you need space for as well. Efficiency, to get this really efficient, to get us doing the 384 samples week in, week out, we've added some automation in. We also need to start working on our top-up strategy for the flow cells that don't quite get to our preferred 30x coverage. So that's going to be coming in soon. Backups for some of these things, like if you get reliant on a robot to do things, it's bad news when it goes wrong. If your shearing machine breaks, that's a problem. So having backups where possible for all of your processes, making sure people can do it manually when the robot's down. Don't forget to order your reagents in time, and we prioritize the blood and the saliva samples during the week so the technicians actually get a weekend off. Consistency is really important to us in this project. It's a very big project, and we want to be able to compare the samples across all the themes, all the sample types where possible. So we've had an awful lot of bioinformatic feedback to fit into this process. Ideally, we don't want to change the process once we've got started. So although we're probably going to hear lots of really cool new innovations from Nanopore, sometimes we just have to say no. We've got where we've got to, and we want everything the same now. And talking SOPs between the labs, making sure, because we are split across 2 labs, that we really are doing everything the same in both labs is really key, which is where the last point, collaboration, comes in. We have weekly meetings between the labs, talking about our problems, optimizing things together, making sure we are doing things the same. We work with our bioinformatics teams, and we work very closely with the nanopore teams helping us as well. So it's, yeah, it's been an interesting time getting it all going, and we're still learning, still building it up, and yeah, top-ups is the next thing we need to learn. So thank you very much, and that's the two lab teams.
Thank you.